dreaming of when I can be part of an amino acid sequence. Then along comes Darwin, a polymerase. Now I'm in an mRNA. Float around through the cytoplasm on the ribosome world land. Oh, it feels like this world is just a dream. 'Cause I just can't wait to be a protein. Oh my gosh! I'm on my way to the process of protein synthesis! I just finished transcription in the nucleus and now I'm on my way to translation in the cytoplasm! Whee! I just can't wait to be a protein! Now we're going through the ribosome, 3 codons at a time. Guanine to my right, uracil to my left. For wasted, we have no time. Look, here comes those tRNAs. Geez, those guys know where it's at. With anticodons on it. Awesome! An amino acid attack. They match up with the complementary codon. Hi, my name's Phil. What's yours? Oh, I just can't wait to be a protein. This is so cool! I'm right in the middle of translation. That's when the mRNA moves from ribosome and the tRNA finds it in synthesis. And, well, on top of the tRNA is amino acids. Amino acids.
What's together for my polypeptide chain?
Yeah!
Let every creature go for broke and sing.
I'm gonna be a protein!
Let's hear it in the herd and on the wing.
Protein!
It's gonna be Simba's finest fling.
Oh, I just can't wait to be a protein! Protein! Oh, I just protein! Oh, I just can't wait to be a protein!
It is the Chef Luigi, and hello, we will teach you how to make your own proteins. Now, here you have, as you can see, we have no cookbook to make a proteins. So where do we get the recipe? The answer is in our DNA. Now this is a DNA. All swirled together, it's called a gene, and it's in this twisty little double helix shape. But in order to get a cookbook, you need for it to go through the process of protein synthesis. Each individual gene sets up an individual protein, an individual amino acid, and that the individual amino acid helps make a protein. So, what you do is you take all these genes and you put them through the process of photosynthesis, protein synthesis, and the ribosome strainer with the tRNA and the mRNA, and you mix it all together, the tRNA and the mRNA, and you mix it all together. Well, actually, that's not exactly how it works, but for the sake of the recipe, I'm running out of time. We must use the ribosome strainer, and you put it through all this, and this is your cookbook. And you use the cookbook to go through the process of protein synthesis. Each little codon adds up to a specific amino acid. You add all these anticodons and codons to make your amino acid chain, and that gives you your protein. Grazie.
The Home Shopping Network. Oh, hello. I did not see you there. Today, I'm going through the best sales of the century. And for starters, we have Pokemon cards. And the best seller of those is the Protein Synthesis Pack. In the Protein Synthesis Pack, you have the basic #1 best-selling card of Protein Synthesis. Protein Synthesis Pokemon card has a picture, HP of 170, and 2 moves, Transcription, RNA polymerase makes an mRNA copy of DNA and sends it into the cytoplasm, worth 70 HP, and translation, which does 100 damage. The mRNA codons are matched with anticodons, and as a result, a polypeptide chain forms. That's a protein, ladies and gentlemen. Next card, we have DNA, which is pretty basic. It has two main moves: replicate, which happens in the cell cycle, and mutate. RNA polymerase, which has two main moves: cut and copy. The RNA polymerase cuts through the DNA and makes a copy of it, where A matches with U instead of T. That's right, ladies and gentlemen, RNA polymerase on sale now for only $80,000. And next, we have our best sellers out of all of them. Out of the whole entire protein synthesis sets, the RNA triplet. If you have the three of these, you can complete any protein synthesis at all. If you have all the other parts. But these are very important, ladies and gentlemen. For starters, we have mRNA. This is a picture. Actually, it's not, but it's the closest that we could find. This is a picture. Actually, it's not, but it's the closest that we could find. The first part is its creation. The mRNA is created in the nucleus as an RNA version of DNA. Beautiful. It's made with the little, where's the card? Oh, here it is. RNA polymerase. It has an HP of 70. And concerning translation, mRNA codons get matched up with tRNA anticodons in the ribosome and make that glorious thing called amino acid chains. Next, we have rRNA in the ribosome. The ribosome is the home of protein synthesis translation, where mRNA is processed and met with tRNA to form proteins. The rRNA processes the mRNA codons and organizes the whole protein synthesis project. They're like the host family of all of this, because ribosomes. And then there's And then there's the tRNA, and they just float around through the nucleus until they find it. A special thing about tRNA with its HP of 70 is its structure. The tRNA has anticodons on the bottom and amino acids on the top, like my beautiful owl hat. It lives in the cytoplasm and enters the ribosome for translation. And during the process of translation, which is another one of tRNA's beautiful moves, tRNA anticodons find their mRNA codon match and make a polypeptide protein chain as a result. Life is beautiful, ladies and gentlemen, and so are the PokeMed protein synthesis cards. Buy the starter pack now for $800,975,2199999 live $6 million. Thank you, protein synthesis cards. Buy the starter pack now for $800,975,2199999 live $6 million. Thank you, and have a nice day. This has been the Home Shopping Network.
What's it like being RNA polymerase?
It's a pretty fun job. I get to cut and copy.
What do you do?
I cut the DNA.
Okay, you cut the DNA and...
Then I copy the NRA. DNA is double-stranded and NRA is single-stranded. A matches T in DNA. A matches with U in ARN.
You cut the DNA! Just say you cut the DNA! What do you do?
I cut the DNA.
Okay, you cut the DNA and... What do you do then?
And then cut the cheese.
No.
It's another level. Oops.
Single-stranded.
No! DNA is double-stranded and mRNA is single-stranded.
DNA double. DNA single.
There's no such thing as CNA. Ben. DNA is... What? How many strands does DNA have?
6.
No.
A matches with T and NRA.
No! Ben, where does A match with T? Answer in a complete sentence.
A matches with T and my angel eye.
No.
Trojan War.
Kate, what are you wearing? No. A matches with...
Pikachu.
No!
mRNA. In mRNA.
I am mRNA.
Wait, no! Puts together for my fully peptide. We are RNA. 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 We are RNA.
Silence! Welcome to the ribosome, mRNA. You are here to be matched with your tRNA codons, and then to become amino acid. Well, actually, you won't become amino acid, but basically, protein shall be made here. What you need to do is you need to split up your letters into groups of 3 codons. That way, tRNA can come. And what are you doing? That way, tRNA can come and attach onto your codons. The ribosome will be the host of that. And that is how this works. Do you like my hair? I like my hair. I'm done talking.